I would like to introduce our speaker for today. Our speaker for today is Professor Shikha Nangia. Professor Nangia is a professor in the Department of Biomedical and Chemical Engineering at Syracuse University, New York, United States. Dr. Nangia received a PhD in chemistry from the University of Minnesota, Twin Cities in 2006 and completed her postdoctoral training at Penn State University. She completed her MAS in chemistry from IIT Delhi in 2000. Dr. Nangia's research uses computational approaches to overcome biological barriers and enhance drug discovery. Her research projects include exploring treatments for Alzheimer's and Parkinson's disease, gut-tight junctions, implantable device-related infections, and epigenetics. Her focus has been to examine the architecture of blood-brain barrier to develop novel strategies to facilitate the transport of drug molecules into the brain. Dr. Nangia's research has received substantial funding to date, including numerous honors and awards for her research, like the NSF Career Award, the Sciloc Fellow, the ACS Open Eye Faculty Award, the ACS WCC Rising Star Award, and she was also recognized with the Chancellor's Award for her outstanding contribution to the student experience and university initiatives at Syracuse University. She is the director of NIH-funded STEAM program that prepares undergraduate students for a career in PhD level biomedical, PhD level biomedical researcher. Thank you, Professor Nangia, for joining us. The stage is all yours.
The title of my talk today is Breaking the Barriers, Shaping a Future Without the Alzheimer's Disease. And the reason I was interested in this research in the first place was the lack of any cure for Alzheimer's. So this is a fact sheet from Alzheimer's Association. And if you look at the statistics on this slide, you would realize that Alzheimer's is the only cause of death in America for which we have no cure. It's the sixth leading cause. It costs trillions of dollars for taking care of the patients and also the families who are participating in this disease and are feeling the brunt of taking care of these patients who really lose the sense of who they are and do not recognize their family. And it happens to seniors so it's an aging disease, and almost 2/3 of the Alzheimer's patients in US are women. So if you keep sort of digging into these facts as a scientist, you realize that what is it about this disease that we do not know, and why don't we have a cure yet? And so along with the aging, it also relates to early death. So if people are diagnosed with Alzheimer's and they are above the age of 70, the morbidity and mortality is higher. So what can we do about it? And if I show you the history of since when we know about this disease, I can trace it back to the 1880s here. And it was a very simple experiment that led to the discovery of this. We will call it the blood-brain barrier, and I will explain why it is a blood-brain barrier. So in 1880s, Paul Ulrich did an experiment where they took a mouse and injected a blue dye, and they observed that every tissue in the mouse body turned blue, whereas the brain and the spinal cord did not stain blue. And later, their student, Edwin Goldman, in 1913, did the reverse experiment, where they injected a blue dye in the brain of a mouse and realized that only the dye was contained within the brain and the spinal cord and not the rest of the body, which led to this realization that the dye is not able to go across this brain and central nervous system, and that led to the term the blood-brain barrier. And Paul Ulrich won a Nobel Prize for this discovery. And this is precisely the reason still that if we look at how much cost has gone into designing drugs for Alzheimer's, it goes up to, say, about 42 billion over the years. And I'm showing you data from 1995 to 2021. You would see that drugs come on the market and there are just 1 or 2 drugs that get approved from the Food and Drug Administration, but they are taken off the market. And we have no substantial improvement in Alzheimer's disease, despite this heroic effort in R&D. So I want to draw this with an analogy that, whenever we have cities and forts and kingdoms to protect, we build castles. Humans build castles. And so here is a castle in Germany, which I'm using as an analogy to say that whenever we want to preserve something precious, we either build high walls or build it on top of a hill and protect it. And there are very specific gateways of getting into a castle. And biology works the same way. So if you look at HIV virus, which is submicron, 0.12 micron here, the capsid protects the virus, but it has a membrane, a lipid membrane around it. So it's a highly regulated interface of what comes in and out of this biological castle, if you would. Same goes true if you're talking about a bacteria. So here is a E. coli. E. coli is a gram-negative bacteria, and it has 3 membranes to protect itself. And the outer membrane is an evolutionary adaptation to protect the E. coli in the various environment it survives in. And we can keep going in size. So here I'm showing you a single cell organism. This is 100 micron in size, and it also has a cell wall, which is highly protective. And if you go to a human embryo, which is only 100 millimeters in size at this stage, it already has multiple barriers. It's a complex barrier. There's a skin barrier. There is an amniotic sac the fetus is in, and there is a gut barrier preventing or regulating what the fetus is feeding on. And of course, there is the blood-brain barrier. It is established very early on. So in all these cases, if you are trying to deliver a drug, if you are targeting a specific bacteria or a virus, there is going to be resistance because it's biologically protected. It's evolution that is protecting it. And so the brain has also adopted this blood-brain barrier as a way to protect itself from any kind of toxins that are circulating in the blood. So if I show you this in a movie here, here is the brain, and here is the brain, and here is a blood vessel carrying blood to the brain, and there are astrocytes which have their podia on the blood vessels and they have receptors where they can take the nutrients in the blood for the brain function. So glucose, water, ions, all those chemicals that are essential for the brain function, they have receptors. However, if you look at other toxins like antibodies or drug molecules that the brain perceives as toxins, those are not transported into the brain. And so the rationale for this is to protect the brain. So if you ingest something which is, which causes indigestion, you get a stomach bug, you don't get a brain bug. So what you have to realize is the brain is trying to protect itself from all these because the motor functions and the decisions that you make on an everyday basis for your well-being are all controlled by your brain. And so when we want to treat Alzheimer's, this blood-brain barrier, which is to protect us, is also a deterrent in treating anything brain-related. So if there is a brain tumor, even now, the only way to do it is to do a surgery to remove the brain tumor. You cannot do chemotherapy. And there are 400 miles of blood capillaries in the human brain. So this highly protective blood vessels are key to understanding how we can treat the Alzheimer's disease. So here I'm highlighting these spaces between the cells in these bright colors to show you that our focus here in our talk would be what is the gap between these cells and what is keeping these blood vessels being different in the brain from the rest of the body. So here is a blood vessel in a static image, and it has all the same features that I was mentioning before. There is a capillary. The cells are making this three-dimensional structure. There are astrocytes, which are binding to the blood vessel and have receptors. But what I want to focus on is the gap between the cells. So if you look at this place called the tight junction at the bottom, that is the area that my research is focused on. We are interested in how two cells interact with one another. You could also see them overlapping here. And what happens here is critical. So if I again take another section of this, now a 2D image, I want you to focus on the green colored oval shapes, which are the tight junctions. These are the junctions that I would like you to think of a Velcro. So if you are in cold and you Velcro your jacket or zip your jacket, this is exactly what's happening between the cells that make the blood vessels in the brain. They really zip up well, or they really Velcro tightly. And that's why the name tight junctions. And they create these very, very specific tight junctions that are not in the rest of the body. So cells are also in our gut, cells are in our kidney and our lungs. The tight junctions in other tissues in our body are much leakier, if you will, than in the brain. They're among the tightest in the brain. And why is it that? So like I said, the brain is going to uptake the nutrients from the blood vessel through these arrows. This is called the transcellular pathway. And in most cases, all the drug discovery that has been focused for Alzheimer's has been how we can get the transcellular pathway to uptake drugs. So nanoparticles people have been thinking about, or monoclonal antibodies, they all target the transcellular pathway. However, this pathway that I'm focusing on is the paracellular pathway. Paracellular means that we are talking about outside the cell, between two cells, that pathway, the tight junction pathway. And what is responsible? So this green oval that I was showing you before, if we zoom in, what you will find is a protein called claudin. It's a member of the claudin family, and claudin proteins are throughout our body. But it is claudin 5, which is highly expressed in the brain, which is responsible for the tightness of this blood-brain barrier. And the word claudin comes from a Latin word called claudere, which is to close. So you can see the primary function of this is to close the gap. And here, the skin-colored box that you're seeing, this is representing the membrane, and it's a membrane-embedded protein. The protein is expressed on the top here, which is called the extracellular part. This is the part which is outside the cell. And at the bottom in red is the intracellular part, which is what anchors this protein inside the cell. And it is the extracellular part on the top here, which is responsible for making this Velcro or zipping up these cells together. Here, and cell B expresses the same set of proteins, that's claudin 5 in a strand, and together they make these tight junctions here. The gap is sealed, and it is this that separates the blood from the brain. So on the top here, if we are talking about the blood, so if I look at this cross section here, there is the blood, and here is the tight junction in green. So how do we know this? So if you look at the schematic here, there are two cells on the left and the right. And these aqueous pores that you see in this schematic here are tiny pores that are left when the Velcro happens, or when the two claudin 5 sections from two sides of the cell come together, they leave some aqueous pores so water can go through. And if you do an electron micrograph from of two cells, so cell represents the tight junctions. And if you zoom into the tight junctions, you would see that the cell membranes are coming together to make these contacts, which are protein based contact and not the membrane sticking together. And if you freeze this cell, so if you freeze these cells and you rip the left side and the will show you that these contacts make these strands. And an example of this would be to think about Ziploc. When you put something and you want to seal it in a Ziploc bag or a container, you press on those the horizontal lines so that you seal the content in a plastic bag. This is exactly how the cells are sealing the contact. However, you should also note that there are some pores here, and that's what we want to target for Alzheimer's treatment. And it's very few and small molecules that can go through. So if you look at what can pass through these tight junction pores that are shown here in the schematic, you would find that less than 2% of small molecules with molecular mass less than 800 Daltons, which form fewer than 8 to 10 hydrogen bonds, can pass through these tight junctions. And I'm very sure you won't be surprised when I tell you which molecules fit this description and are able to go across the blood-brain barrier. It is alcohol. That's why people get addicted, and that's why it makes an effect, an instantaneous effect. It goes, if you consume alcohol, it goes in your bloodstream and crosses the blood-brain barrier. And that's why you lose your motor skills. Nicotine, we get addicted to that. This is a small molecule that goes through. You can see it's small in the size. And then also caffeine. We get a caffeine fixes every morning. That is also another molecule that passes the blood-brain barrier. So things that humans have either for their benefit or for getting intoxicated, plus or the minus, there are molecules that go through, and that's how the blood-brain barrier function works. So the overarching goal of my lab is to study the therapeutics that can cross the blood-brain barrier. So there are multiple claudins that are we are using computational modeling to understand these because there are no experimental instruments available to understand what we are trying to look at experimentally, because we do not have the resolution. All these pores, you will see in a little bit, I will show you, are less than 1 nanometer in pore size. And membrane proteins experimentally are very hard to crystallize, very hard to isolate. And here you are not only dealing with membrane proteins, you're dealing with an array of membrane proteins between two cells. And the character that you're looking for is between the two cells. So experimentally, it's an impossible experiment at the moment. We will have to come up with a very sophisticated instrument to look at it. So this puts computational modeling in the forefront, and we can really resolve these things at an angstrom level using the techniques that we are using. And I'll share some of those with you. So what have the advances been? And I will show you one of them. So breaking the blood-brain barrier is not impossible if you actually do some microbubbles and an ultrasound, as shown here, in a small section of the brain. What will happen is you can mechanically break the tight junctions, and you would see it here, the cells are moving. And what is happening in the process is you have mechanically broken the tight junctions. You may be able to deliver a drug that was injected just prior to the ultrasound. But what happens is in the process, you are also exposing the brain to the toxins. So it is not acceptable to just break the tight junctions. You also have to recover them because they are important for the normal functioning of the human body. Amplified form of this would be a traumatic brain injury. When somebody has a head injury or has a concussion, what really is happening is the breaking of the tight junctions. That's why the blood gets into the brain because the tight junctions are broken, and that is fatal in many cases. So when this experiment is done, it has been done in mice, and it has caused detrimental consequences of this ultrasound. And so this is not a viable way to treat Alzheimer's or deliver drug to the brain, because the tight junctions do not recover as easily as they are being broken here. So now I will delve into what we do in our lab. And for that, I have to acknowledge my research family, family of PhDs, undergraduates, master's students, some of whom are shown in pictures and mentioned in the list. Most of my students have actually been working for big pharmaceutical companies here in US, and you can see the names next to the PhD students where they are. And also my collaborators, because when we do computational modeling, we want our collaborators to verify in the experiments what we are doing. So I have a rich collaboration with many also at Syracuse University, listed here. So how do we do it? So let me just now give you an overview of how simulations work, if you're not familiar. Computational modeling, depending on what question you're trying to ask, what resolution you want to ask. So you can go all the way from angstroms to micrometers if you're interested, or if you're interested in processes that happen at a picosecond time scale to over seconds. You can choose the level of granularity you want. And for each level, there are different methods which are listed on the right. So if you're interested in where the electron cloud is and where the nucleus is and what is the role of the solvent next to this, you could do quantum calculations. If you make an approximation that an atom is a hard sphere and has a certain radius, and you could perform an all-atom simulation, or if you cluster a few atoms and call them as beads, then you can say it's a coarse grain simulation, and so on and so forth. And you can imagine that as you go from one level to the other in granularity, you are going to make some approximations. And depending on what level of detail you want, or what size of the system you want to simulate, level of approximation, and of course, verify against experiments. And how does this all work? It's based on a very simple principle, and I will use the quote from Richard Feynman, who got Nobel Prize in 1965, who said, everything that is living can be understood in terms of jiggling and wiggling of atoms. So if you are able to simulate the jiggle and the rotations and the bombardment of atoms with one another, and you see the torsions and all these jiggling and wiggling motions of atoms for a prolonged period of time, and if you could afford that, you should be able to understand how the processes are working in biology. And here is a schematic of, or a cartoon of how, I want to show you here jiggling and wiggling. So if you have a drug molecule in the center shown in cyan, and you have water surrounding it, you can see the hydrogen bonds in yellow getting formed and broken, and how much this drug molecule is interacting with water or not. But to get all these pieces of information correct, there are a bunch of equations that go into this. We call it the force field. And it takes into account the bonded terms, how long are the bonds between the atoms, what are the angles between the atoms, what is the torsion in the dihedrals, and also the non-bonded terms. So when water is interacting with the drug molecule, it's not bonded to it, but is it electrostatically attracted to it if there are charges? So that's this term, or it's just the Lennard-Jones description of how two molecules are stable when they interact with one another, or repel each other when they are together. So for example, oil and water would have a very different Lennard-Jones term here, the 12 and the 6 potential, as it's called, versus say hydrogen bond, where if you have a electronegative atom, then the hydrogen bond will form with that, with water. So all these numbers that go in this equation have to be precise, have to be checked, and also many of these are derived from experimental data to get the right description of what you're trying to study. And getting this to work is the primary prerequisite to get a simulation right. And if any of these are incorrect, we say it in computational word, garbage in and garbage out. So if you get the angle of a water wrong, the output you're going to get is also incorrect. So you have to be very careful at this stage. And so this is one of the important factors in running simulation that we do, that we are using reliable parameters or descriptors for our molecules that we are interested in. And this has been going on for 3 or 4 decades now. So there are reliable parameters available for most of the biomolecules, and that's what we use in our work. So here I'm going to show you now the first time when we started doing simulations. Here is a membrane, because I told you Claudin-5 is a membrane-bound protein, and it is sitting in this lipid membrane made by these blue dots, which are the lipid head groups, and the tails here, and the water is shown in pink beads. This is a coarse-grained simulation. It represents 1.1 million beads, which means this is about 5,000 or 6,000 atoms. And it's a patch of a membrane, which is 60 nanometers across 60 nanometers. And I'm going to show you now what I mean by jiggling and wiggling. And so I'm going to show you in a small patch of the membrane here what happens. And you're looking at the Claudin-5 proteins from the top. And as I start my simulation, you would see that claudins are finding each other in the lipid membrane, and they're sticking onto each other to make dimers, and the dimers come together to form a larger strand. And this process will continue. This is 1.1 million beads, like I said, and it takes a computer almost 80 days to get this assembly right. And I'm going to show you in a moment what the strands look like. And this work was done by Jerome Ridunathan, who is now graduated and works for Genentech. These were our early days when we had just started doing simulations. And here I want to show you that if you start with a well-separated array of claudins, shown again in pink, in a lipid membrane, and you're seeing from the top, you would see at 5 microseconds, dimers and trimers have started forming. And if you let it go to 10 microseconds, you start seeing strands. And if I zoom into this, one of the strands here, and I say, okay, these are beads, let me look at the atomistic level, how this is interactions are happening, because that's more detailed. What we see is that the claudin proteins interact at specific orientations. And we start looking in, what are the specific orientations? And what we found is that these proteins can either interact. Now, these are the colors I showed you in the beginning. There are 4 passes in this protein. And dimer A is a specific orientation in which the 2 neighbors come together. B looks very different from A. And in D, for example, the 2 proteins are very far. They're only talking from their extracellular loop. So what we found is that there are different ways these proteins come together. And an example would be that if we were all, in a big stadium, and we say to all people, line up, and don't tell them that you have to stand in arrays, most of us will stand in different orientations and make a crooked curved line, depending on who we know, who you are with, and who we are having a conversation. So, you could be shoulder to shoulder, you could be back to back in a human row of, a human row. And that is exactly what proteins are doing. So they have specific ways of coming together, and they are not becoming a mob of people, if you will, in that sense. And so we actually looked at experimental data, that are we simulating it correctly? And what we found in the literature through FRET analysis and various other claudins people had studied, that people had reported dimer A for a specific claudin, may not be Claudin-5 in the brain. And there were examples experimentally, people were reporting orientation. And none of the experiments ever said that you would need a collection of A, B, C, and D to really make the whole strand. And it is similar to this analogy that, the blind man and the elephant story. in which if you are blindfolded and you are looking at a specific part of an entire elephant, you may not realize it's an entire elephant because you're looking at a specific part. And that's what experimentally was happening is people were reporting a dimer, but they were not seeing the big picture that when you have all these together, it makes a strand. So this was very insightful from our first work that we did. And then we said, all right, this is a two-cell system. The two cells have to come together to make the tight junctions. And so here what we did is we set up two membranes on top of each other, shown here. Both membranes have claudins. Water is the same orientation. This is a bigger membrane than before. It has 2.4 million beads. And when we ran this simulation, we started seeing that the top layer here and the bottom layer, the claudins from both the cell membranes started to interact with one another. And if I strip away the membrane or the water here, and I rotate this output 90 degrees, what you see is in the right. It is the same orientation now to show you what I described to you in the beginning, the kissing points that were occurring. And this is, again, a result of proteins interacting. So the kissing point that you see here is the kissing point that you see in the picture here. That was a result of the simulation. However, these are very expensive simulations. They can take up to months, and PhD is a very critical time for a student. And if one simulation is taking 3 months and you have to do repeated simulations, the analysis becomes cumbersome. And so we said, how do we reduce the cost of this simulation? Because we cannot keep doing these simulations for weeks on end. And what is a strategy? So necessity is the mother of invention. Computer algorithms are getting smarter. We said, can we develop a method which can give us the output of what are the key orientations of the dimer without spending this much of time? And so here comes the advantage of being a computational scientist, because you can create your own tools, which are much more difficult and much more expensive experimentally. So I would like to emphasize that computational modeling is a growing field, and with quantum computers in the horizon, this field is going to explode in terms of its capabilities. So I encourage you to think about this. If you're a student who's thinking about PhD, computational modeling, in whatever, whether it's energy or biology, you should be thinking about this field. So how did we overcome the challenge of the computational cost? Because it is, a long time it takes the simulation. So what we said, all right, so our protein is embedded in the membrane, and it can only take a few rotations around each other. So if my protein A on the left or protein 1 on the left, and the arrows that you see, the blue, red, green, and yellow, are the 4 TMs, the 4 transmembrane loops, because remember, this protein goes through the membrane 4 times. And we said, this protein, relative to its neighbor, can rotate 0 to 360. It's stuck in the membrane. All it could do is a rotation. And if the other one, protein 2, can also do these rotations, then you are sort of turning 2 knobs together, 0 to 360. And that says there are possibly 129,600 orientations with 1 degree increments that these two spheres or circles can take with respect to each other. So this is my rotational space. I want to figure out how the blue-blue interaction or the blue-green interaction, which is more favorable. And so we said, instead of this huge membrane of 60 nanometers, what if we just take 2 proteins in some random orientation relative to each other and just put it in a small patch of a membrane, which is very affordable to do, and actually break this space up from 0 to 360 on the x-axis and 0 to 360 on the y-axis into bins. And every dot that you see here in this bin is a point that could be a simulation in its own. And so here is sort of a movie showing, if we just do 2 claudins, how do they interact with one another? And I've taken away the membrane here. And if we do this simulation in many small pieces, so if I break this space up and every dot is a separate simulation, we have computational resources to break this up, and every dot does a dimer, and it's randomly oriented, and you can see from where it originates, there are different rotations. And we run these simulations independently. So if you take this dot as one of the starting points, and as MD happens, it rotates around it a little bit. So all these little dots are the places or the orientations the dimer took. And we are running these thousands of these simulations. So what happens is you can get lots of data in this space, and every dot is providing you the orientation, the energy, the stability, and you can keep doing this. And what you end up doing is, instead of the 29,600 grid points, you actually get millions of data points because you visit every orientation several times. And from this data, millions of data points, what you get is a potential energy surface, which tells you the deeper the well, the darker the color, the more stable the dimer, the brighter the color, yellow the color, it is not stable. So now you can tell that A, B, C, D dimer that I was showing you before may not be all the dimers. There are many more. There are many more valleys here. And you can pick with confidence that which one is the most stable. And cost-wise, which was the whole point of this development of this method, which we called panel, and Nandini Rajagopal, who is also in Genentech, was the student behind developing this method. And you can see the advantage of this method in terms of it's an order of magnitude lower. Instead of 81 days, we can do these simulations now in 3.4 days. It's highly automated, and actually it can be applied to any membrane protein. And this was on the cover of JCTC a few years ago in 2019 when we developed this method. So with this method now, we are confident what dimers come together, and here they are numbered. 1 is the most stable, 2 is the next stable, and so on. And using these, what we can do now, we can figure out every valley which has that orientation. And using these orientations, we can say, all right, so these are the dimers that are stable in cell A. Cell B is also going to have a similar dimer, and let's see how they come and make this Velcro, or how they come together, and see what kind of pores they make. So the output here was, out of these combinations, there were 2 orientations that create pores. And if you look through this pore architecture, you would find that there is a pore here, which is in diameter, in the most constricted part, 0.85 nanometers. So it is smaller than a nanometer. And here is the path a molecule will take. So if you have a caffeine molecule or an ethanol alcohol, this is the path it will take, and it's able to go through. And this result was reported in one of our papers. And there is also data that these pores are very selective. They would obviously allow water to grow through. So if you see the hills and the valleys in this blue curve for water, you would see there are not many hills and valleys, so water can really go through. But if you see the barrier, or how high in kilojoules per mole the free energy is to move a sodium ion or a potassium ion, it's really high. Chlorides can go through because at the beginning or the mouth of the pore, the barrier is high, but orientations change it and the barrier goes lower. So chloride ion can go through. And here, if you compare the divalent magnesium and calcium ions compared to potassium and sodium, you see the barriers are much higher than water. And here the scale goes up to 18, here it goes to 30. It tells you that the brain is definitely not transporting positive ions, especially calcium and magnesium in the brain. And this result was on the cover of Langmuir earlier. And this showed for the first time what Paul Ulrich was mentioning. The blue dye did not go into the brain, too big to go through because of the pore size, because of the selectivity. So we've been studying various other Parkinson and Alzheimer's related drug molecules to see what is the point where they cannot go through, where do they get stuck, what do they interact with in our effort to design drug molecules that can go through the tight junction. So if this is our probe molecule in blue, this is the tunnel or the passage, which of this pore, so that drug molecule can go through, but we do not want to break this pore because it's important for blood-brain barrier. So in collaboration with my collaborator at Rensselaer Polytechnic Institute, what we are saying is that if it's cell A on the left and cell B on the right, and these are the extracellular loop 1 and 2 that are interacting to create the tight junctions, which I just showed you, can we design small peptides called pepdarts, which stands for permeability enhancing peptide for the disruption, attenuation, and the recovery of tight junctions, so that you can poison, if you will, the ECL interactions. So the two cell A and B are not so tightly bound to each other because the peptide is screening that. And you make the drug molecule go through because you've increased the pore size here. And then the peptides have easy binding and unbinding events so that you recover back the tight junctions. So that is our strategy. There are ongoing in vivo experiments based on the peptides that have been designed, and I do not have the liberty to share with them because it's still under testing. But what I would like you to emphasize here is that if you understand the structure, if you understand why the blood-brain barrier exists rather than the output that, oh, there is a blood-brain barrier, if you understand it, then you can design drugs for it. And the transcellular pathway of getting through the cell is much tougher and has not been successful after many, many years of trial. And this paracellular pathway between two cells, which is the analogy of the Velcro or the zip between two cells, opening that up to regulate the size is a more viable option, which we are testing both in simulations and in experiments. So to sum it up, I would go back to this slide, which I started with, that yes, we have castles. Our brain is a castle, it's a biological castle, but there are specific pathways that lead you to the inside of the castle. You got to know where to enter